Thanks very much for having me, Peter. This is a really cool format, and I've been looking forward to it for the last couple of weeks. And this work really focuses on AGRP neurons, or a small population of neurons in the arcuate nucleus of the hypothalamus. They're genetically defined by the neuropeptide AGRP. So they're unique in the hypothalamus and indeed in the brain in expressing this neuropeptide, which makes them genetically easy to manipulate. And also because of their proximity to the median eminence, they lie at least partially outside of the blood-brain barrier and thus can sense nutrients, metabolites, and hormones in the blood in a way that other neuron types in other brain regions cannot. And people have utilized their expression of AGRP to genetically manipulate these neurons for close to 20 years now to show that these neurons are absolutely necessary and sufficient to drive feeding. In 2005, Richard Palmiter's group showed that by ablating this neuron population in adult animals, you could make a mouse starve to death in about 5 days. Not only would these mice not eat, but in fact, if you gavage them with food, they'll actually reject the food and die anyway. Conversely, in 2011, Scott Sternsen's group and several other groups showed that stimulating AGRP neurons using optogenetics was sufficient to drive feeding, even in a sated mouse. Experiments like these two genetic studies, coupled with the location of AGRP neurons and a large number of other behavioral, physiological, and histological studies, grew into a model of AGRP neurons as detectors of nutritional state, kind of passive detectors of nutritional state, whereas levels of nutrients and satiation hormones dropped during a fast, these neurons would become more active and drive food seeking, and then as you ate food, nutrients entered your bloodstream, metabolites entered your bloodstream, and satiation hormones began circulating. Their activity would gradually come down over the course of the meal, and ultimately you would stop eating. But it turns out, once calcium imaging kind of became widely available, Zach's group and several others showed that, in fact, a major regulator of AGRP neuron activity in vivo is actually food cues, or the anticipation of food intake. So that's shown in this figure. When you present a mouse with what we call caged chocolate, which is chocolate under a little wire basket that the mouse can see and smell but not actually eat. I think it's the meanest experiment I've ever done to a mouse. Their AGRP neuron activity drops off precipitously within seconds. But if the animal can't actually follow up that sight and smell of food, or that knowledge that food is available, if they don't follow that up with actually eating, if you leave this chocolate caged, then AGRP neuron activity rebounds. The animal doesn't forget that it's actually hungry. Its AGRP neurons become active again. And then when you uncage the chocolate, the AGRP neurons again drop their activity very rapidly. But this time, as the mouse eats the chocolate, the activity of AGRP neurons stays low. And what I was really interested in studying is what's the nutritional aspect of this? Why does this activity after caged chocolate bounce back up and this one stay down? And unfortunately, that's not something that you can study in a freely feeding mouse, because in a freely feeding mouse, the second it sees or smells food in the environment, or even sees a neutral cue that it has come to learn predicts food, you lose the whole dynamic range of activity of AGRP neurons. I developed this model for simultaneous intergastric infusion of nutrients and recording from AGRP neurons to show that infusion of nutrients directly into the gut is sufficient to inhibit AGRP neurons. Water does nothing, and basically show that while exterosensory cues like the sight and smell of food are sufficient to inhibit AGRP neurons, that nutrient intake is really necessary to stabilize this inhibition. And in fact, nutrients themselves are sufficient to induce this inhibition, albeit over a slightly different time scale, minutes over seconds. This inhibition is independent of macronutrient identity. So glucose, peptides, fats, whatever we infuse cause a similar amount of inhibition, and the magnitude of this inhibition is proportional to the number of calories infused. We went on to show that a subset of gut hormones are sufficient to inhibit AGRP neuron activity on a time scale consistent with fast refeeding or with a meal. I'm highlighting CCK here because the gut hormone cholecystokinin we showed is actually necessary for lipid-mediated but not glucose-mediated AGRP neuron inhibition. And without belaboring it too much, we developed this idea that while all nutrients can inhibit AGRP neurons when delivered to the gut, the mechanisms by which they do so at least appear to be macronutrient dependent. Along with trying to figure out and dissecting the mechanisms of how glucose or carbohydrates are inhibiting AGRP neurons, how peptides are inhibiting AGRP neurons. One of my big interests was in how disease states, and particularly obesity, might modulate these dynamics to help promote weight gain or to try to pull an animal back towards its homeostatic normal weight. And so to test this, we used a paradigm which is depicted here. We basically took lean mice on a normal chow diet at baseline, recorded AGRP neuron activity in a bunch in response to nutrient infusion or satiation hormone injection, repeated this after a period of 6 weeks on a high-fat diet, and then allowed the mouse to lose weight over about a month and repeated them one more time to assess the changes. And what we found is that a high-fat diet-induced obesity attenuates the AGRP neuron response to dietary fat ingestion, and this is nutrient-specific and not reversed by weight loss. So over here on the left are data from a group of mice that are maintained on standard chow diet throughout the paradigm. And you can see they maintain a really stable response to the intragastric infusion of lipid over the 10-week paradigm. By contrast, the diet-induced obese animals after 6 weeks on a high-fat diet have an attenuated response to lipid infusion. And after 4 weeks of weight loss, you can barely see it back here, but the inhibition, this attenuated response is maintained even after a significant amount of weight loss. By contrast, when we infuse glucose or peptides in mice at baseline or after 4 weeks on or 6 weeks on a high-fat diet, the responses remained relatively stable. We also showed differences in the response of obese mice to extrasensory cues and showed differences in response to some of the hormones that we'd given previously. And Mike Crash's group has a really wonderful paper that also just came out showing some of the same things. The kind of take-home message is that some of these sustained changes in neural dynamics may contribute to difficulty with weight loss and may represent kind of a neural correlate of this elevated set point that we think obese people may develop. There are obviously many, many, many next directions to go, but a couple that I think are particularly interesting are, number one, we've shown that dietary fat communicates with the hypothalamus via the CCK and vagal pathway, and understanding how carbohydrates and also how proteins communicate from the gut to the brain, I think is a very important next question, and I'm talking about like on the molecular level. And in terms of glucose, there's been some very cool new work by Diego's group and then by Charles Zuger's group. The other direction that I think is really, really interesting and overlaps with my clinical interest in obesity and insulin resistance is to understand, we showed that high-fat diet selectively alters fat responses. I think the question is, does overnutrition with different macronutrients elicit distinct changes in gut-brain dynamics? And could or should this shape obesity management, either pharmacologic or nutritional? And it's something that I look forward to seeing how it evolves over the next several years.